So following our last analysis round, I placed the parameter file outside of our batch of test images here. But again, that just captures the information from our experiment. So FOCUST allows you to save optimization files when you're still configuring your segmentation parameters. And it also allows you to save a parameter file, which is automatically created every time you run an analysis mode. And remember, analysis modes allow you to batch process images, and they apply special relationships between object types that you define. So if we open up our Analyze window again, say that I have come back after the Christmas break or the holiday season, and I've just generated some new data, which we will hypothetically say I've added into a new folder here. And I would like to rerun that analysis on this new data so I can continue to build my sort of master data sheet. Instead of having to re-enter all my parameters again, or forget what variable names I used or analysis mode for my experiment, I can load a configuration, but this time, instead of selecting the optimization mode, I'm going to select the parameter mode from my previous analysis run. And when I hit open, we can see not only that all of the segmentation parameters have been copied across, but so have my preferences about the tertiary objects or my grouping variables and the analysis mode as well. All I need to do now is point focus towards the image directory that I'd like to process. And we're ready to go. What I'd like to show you now are some of the additional label processing options that we've included in FOCUST. It's very 3D focused, as the pun suggests. And so one of the things that I was interested in was the spatial distribution of stains within cells, within tumors or spheroids, and within nuclei in the case of speckle mode. I was also interested in the branching morphology of interesting shapes of cells and indeed nuclei. So we have this checkbox we said we'd talk about later. Well, it's later now, so let's see what happens when we open this menu up again. Again, we have options for primary, secondary, and tertiary object modification. And we have three options in each box: 25 or 50% bands, or an option to skeletonize. Now, this allows us to add some additional processing steps to any of our object types, regardless of the bio mode that we choose. Okay, so in this case, we're still using single cell mode because we're using the same example images. We have our segmentation parameters set up exactly as they were, but this time, let's say that I would like to know where inside of my cell the nucleus sits spatially. Is it close to the middle of the cell or is it right towards the edge? Let's say that I also would like to know how many branches each cell has across major axes. This is the kind of data that I can extract using these three options. If I select 25% bands for my secondary objects, let's say, my cell shapes will be broken up into 4 bands, so 25% of my cell per band. The same for 50, so I can generate a core of my cell and a periphery of my cell. Let me show you what I mean. Let's go to the documentation. Let's scroll down till we see label processing. In FOCUST, we've implemented Skeletonized 3D and Analyze by Ignacio as part of the standard FIJI distribution now, which allow us to selectively skeletonize shapes in 3 dimensions and interrogate the morphology of those sort of major defined axes there, which is really cool. Interestingly, in testing, we noticed that a cell would be given one label and its skeleton would be given a different label. And so because we wanted to take care of the data handling, and we obviously for single cell node need to match up a cell and its nucleus and its cytoplasm, and now with its skeleton, all skeletons that are generated in single cell mode, and indeed in any mode, are actually label matched back to the original cell that they came from. So we confirm that the skeleton that we assign to that particular row of data is indeed produced from the correct label. I'll show you what I mean shortly. This is really cool because it gives us some awesome insights into morphology and again allows us to correlate that with things like volume or stain expression, etc., etc. What we mean by bands, if we scroll down slightly, we have this process called stratification. I was very interested in figuring out where my nuclei sat inside of a tumor. And so I developed this method to break apart a label using distance maps and some thresholding. If you select that 25% band box, your shapes will be broken up into quarters. And in the data set and in the files, we append that with a Q, where Q1 is the core of that shape, Q2 is the next quarter, 3 is the next quarter, and Q4 is the outermost component to that shape. And we can see that when we combine all of those regions together, we've broken this tumor into 4 distinct bands based on their distances from the periphery to the core. And we can see how that looks in a 3D rendering of this particular shape. We have indeed broken that shape into 4 distinct zones. This gives us some awesome resolution to have a look at where a stain inside that tumor is more strongly or weakly expressed. Gives us the ability to count how many nuclei occupy each of those distinct three-dimensional spaces. So we can see if lots of nuclei or lots of speckles are more core oriented or positioned inside of the shape, or whether they are in fact all peripheral. These are the kind of questions that I wanted to answer in some of my other research. And so this method is something that I'm quite proud that we're able to include in FOCUST. As we said before, 25% bands get the prefix Q, where we started in the core and move outwards. And if we pick 50% bands, we go to halves, so H for half, where again 1 is the core and 2 is the periphery. A note on that, if you have objects that are touching borders, it gets a little bit weird and doesn't really work as well. So just make sure that you're using either objects that are well sampled and so you have plenty of padding above and below your shape, or you select the correct kill borders options to avoid any funky distance maps. So let's have a look at what this process looks like. We're working with some single cells still, a SAM dataset, and I would like to know where inside my cell the nuclei are sitting. Are they closer to the edge or are they more in the middle? Now I could just tick 50% bands to break my secondary objects or my cells into a core and a peripheral band. But I kind of also want to tick the 25%. It's not an either or. You can select both and then explore the data after you've generated it. For my cell shapes, I would also like to know what sort of major axis branching, what sort of funky morphology we've got, and so I'm going to tick skeletonize for these as well. Skeletonize and banding should really only be selected on full or complete objects. So because our tertiary object is a cytoplasm and it has a great big hole in the middle that represents where the nucleus was, it would be inappropriate to select these options for tertiary because they will be malformed and the data will be fallible. And I don't really have any good reason to explore the distribution of a stain in my nucleus itself, nor do I have a good reason in this case to look at axes of my nucleus either. Great, so for each cell, we should generate some 25 and some 50% three-dimensional bands or stratified areas, and we should also look at the skeleton of those shapes. Let's click run analysis and explore some of the outputs that we get. Again, we get that parameter file generated, which this time will also copy over the options that we've selected for 25% and 50% bands as well as our stratification. In our log, we now also see some additional things appearing around measuring band intensities and skeletonization. and all kinds of different things. We get reports on how long it took to stratify and quantify a particular set of shapes. I think that was image 1 just finishing there. And we can also see in our outputs that we have a large number of images now, actually encroaching on our space here. So notice that we now have secondary objects Q1, Q2, Q3, and Q4, as well as secondary objects H1 and H2. And that's because we selected both 25% bands for our quarter breakdown of the shape and our 50% bands for the half breakdown of our shape. Remember, 1 in both cases is the innermost component to that stratification. So core is H1 versus periphery is H2, whereas core is Q1 versus periphery is Q4. We also have some skeletons that were generated from our secondary object, again because we ticked the box. And in full transparency, FOCUST outputs every image that you have selected so that you have the ability to go back and check your data and really understand the outputs that you're generating rather than just blindly looking at numbers in a CSV file. So before we open the CSV file, because there is now a significant amount of data in this that we should talk about, let's first have a look at maybe the skeletons. So let's have a look at the skeleton file. And as we scroll through this, we can see that there's definitely some malformed skeletonization going on here, again from inappropriate shapes that we were trying to skeletonize. But we see these winding tracts through the axes of our shapes and the branches of our shapes. And it's probably easiest to visualize this if we make a Z projection of our image. And we can now see that we've generated major axes and sort of branching artifacts from all of our shapes, which is pretty cool. So for example, if we were to open up our secondary image, or sorry, our secondary objects file again, where'd it go? There it is, for single cells 1. We project this image as well. We can start to match up some skeletons with some shapes. So this skeleton here, this cell here. So we start to be able to explore some really interesting cell morphologies. And of course, if they're imperfect shapes, or there are holes in those shapes, or there are problems with segmentation, absolutely the skeletons will be a bit rubbish. But it's the potential for extracting some really cool data here is very high. So we have some skeletons, that's cool. Let's have a look at some quartiles. So I'd like to open up Q1 for cell 1, Q2, Q3, and Q4. Let's open all of these up together. And it looks a little bit funny because these shapes are very, very small. But hopefully what you'll notice is that Q1 represents the core of each shape, while Q2 represents the next band in, right? So if we find something kind of in the middle-ish, maybe here, try and find the matching shape. There we go. So we've got the outermost band of this cell and the innermost band of this cell. And again, these cells are very, very small, and this is a distance-wise calculation. So using 25% bands on these shapes is really not the best idea because they're just too small. There's not enough pixels to represent the space. So I'll show you this on some spheroids in a moment. But let's have a look at the data. So again, we have our single cell name for our images. It's all stored there for us to have a look at. We've got our labels. We've got our indexing for the labels for cases where multiple nuclei were found inside one cell. So cell 3 contains 1 and 2 nuclei. And again, we've got information for our nucleus because that's the primary object. And as we scroll across, we then run into the columns that pertain to the secondary object or the cell. And as we keep going, we will eventually run into, again, we've got our data here, our intensity information for the secondary object. Then we run into, at the end of our intensity information, secondary skeleton number. So this is now the skeleton ID that has been matched back to the shape of the cell. And we have a number of metrics here. We've got sort of the number of branches, the number of junctions, endpoint voxels. All of this information is available from the Analyze Skeletons plugin that's linked in the documentation. And so you can see the maximum branch length as well, which is really cool. If we keep on going, and again, everything in this row pertains to the same cell. So the skeletons are label matched back so that a single row still represents one biological entity. As we keep scrolling, we run into some more intensity data, but this time it's appended with the stratified region that it's been extracted from. So if we find the mean value, because that generally makes the most sense, stop it. There we go. So we have Q1, which is the quarter of the cell. So the innermost portion of this cell, this is the mean DAPI intensity. Okay, so all of that intensity data again is stored at a single cell level, and it's all row matched. So as we work our way across, we then run into Q3 eventually, and then Q4, the outermost component to those cellular rings that we've created, and now we're at H1, which is the halves. So we store the skeleton data, the quartile data, and the half data for the shape, all in the same row. So it all pertains to the same cell. So we end up with a fair bit of information all the way over at OL in Excel. But it's then incredibly well positioned to go straight into either in Excel or something like GraphPad, Python, or R, where you can then start to correlate and extract some really, really powerful insights on your 3D matched data. I will now, through the magic of editing, cut to some spheroid stuff to show you a proper interactive version of what that looks like. Okay, so if I would like to do some analysis on some spheroids, just as a demonstration of our additional label processing here on some slightly bigger shapes so the distance maps actually look half decent, we'll go through a similar process. We will go and point our analysis mode to our spheroid dataset here, where we have two spheroid images. I have previously optimized the segmentation for these, but let's go ahead and just enter some stuff here. So just so we can have a look at the data that we're actually going to be working with here, let's open up one of these and have a quick look so we know which channels we're working with. I've gone ahead and optimized the segmentation for these ahead of time, but let me just change our lookup table here so you can see it slightly better. So channel 1 here are nuclei through this spheroid or tumor in the z-axis. Then we have a protein of interest, which is mostly nuclear localized. Then we have a protein of interest which can shuttle in and out of nuclei, or a transcription factor, I should say. And then we have phalloidin stain again, or the filamentous actin network throughout our spheroid, or our mini tumor here. So that's the data set. Let's go ahead and I'll just quickly configure our segmentation here. So I know that I need a Gaussian blur. I don't need a secondary filter, and I'm going to go ahead and plug in some values that I had prepared already, and I'll leave that as Otsu. Great. And unlike the single cell dataset we've been working with, our channel that represents the whole tumor or the whole spheroid is in channel 4 with that phalloidin stain. So I'll go ahead and configure the segmentation for this guy as well. Except here, because I only have one spheroid per image in this data set, I'm going to change from maxima to connected component. And that's going to join all the pixels together that are touching. If they have a neighbor, they'll be given the same label. So instead of having to partition objects from each other with some of these other segmentation methods, connected component allows us to just join pixels together from the outstanding library of mathematical morphology that is MorphoLibJ. Because of that, I don't need to change any of these size parameters here. Whatever I put in there won't be used anyway because this is a separate algorithm. And I am going to manually define a greater constant here as well. Let's go ahead and add a grouping information. You don't have to have underscores. Let's just show that you don't have to do that. Let's go ahead and not add channel names here, so you can see that when we don't add channels, we default to C1, C2, C3, C4. And instead of single cell mode, let's use the spheroid mode. This also now allows me to select my label processing. I don't want to do anything to my nuclei, but I would like to stratify my spheroids. So let's turn them into 25% and turn them into 50%. And let's process. Okay, so that took a little bit longer because these are larger images and we'll be doing some reasonably complex analysis. So again, we get a full log file with a breakdown of all the time components to this as well as a notification that our batch processing has complete because we did process 2 images. So we had spheroid 1 and spheroid 2. And these note are in a raw Nikon file. So we can process different image types natively in support inside FOCUST as well, thanks to FIJI and the magic of bioformats. So let's close this, and then I'll also close our analysis pane there as well. Note that we have run in spheroid mode. So we had spheroid mode ticked, recall that, which means that we have different results outputs. So as the documentation said before, if we look at our analyze mode and then a breakdown of the different modes, previously we were working in single cell mode, and that said that we could manage multinuclei and all data matched into a single results file. Whereas in the spheroid mode, we actually just relate nuclei to spheroids, but we keep those data sets independent of each other. So we match them by figuring out which parent nucleus belongs to each spheroid and vice versa. Sorry, which nucleus belongs to which parent spheroid and vice versa. And we also, when we do stratification, calculate the number of nuclei centroids per stratified band. So if we now have a look at this data, we open the primary objects data file, we can see again that we have our grouping label if we would like to include one. Again, if we don't have one there, it will not be written. We also have the image ID that's stored there too. Let's just quickly have a look at our primary objects for spheroid 1 here as well. And we can see that we do have some nice sort of nuclear segmentation throughout the spheroid there. Back down again, cool. Doesn't look too bad. So let's have a look at the layout of the data here. So instead of primary dot volume, because this is just the spreadsheet for primary objects, this is just volume for the nuclei. So these are just the nuclei inside of spheroid 1. You can see as well, we do have some very, very small nuclei, a single voxel in fact, so I would argue that's not a nucleus. So some preparation of this data is probably needed before we go ahead with some analysis. But as we scroll across, again, remember we left our naming variables for channels empty. And so instead of taking a variable name, the CSV now just contains C1, C2, C3, etc. When we've done intensity quantification inside these shapes. So when we go all the way across at the end of our data set here, we get some cool insights into where these objects are situated in 3D space. Okay, so because we ticked stratify secondary objects, and remember secondary object in this mode is the whole spheroid, whereas primary objects in this mode are still nuclei. Because we stratified the whole spheroid, we get a record of where each nucleus, which in the primary objects datasheet is each row, one row equals one nucleus. We get a record of which parent spheroid that came from, so spheroid ID 1. And then we get a breakdown of each band. And where we see the label of our nucleus inside a band, that means it was found inside that space. So we should only expect to find a nucleus inside one quartile band and one half band, because remember we ticked 50% and 25%. So Q1 is 0, so it's not there. Q2 is 0, it's not there. Q3 is 1. So nucleus 1 was found in band 3. That's the arrangement of this. Okay, and if it's found in band 3, you would therefore expect that it should be in the outer half of a spheroid. And indeed, when we look at the halves, remember H1 is the core, H2 is the periphery. It's not found in H1, but it is found in H2. Okay, so having the extra resolution to know, is it right at the very edge of the spheroid, or is it a little bit further in? We think that that's pretty cool. So if we then have a look at the secondary objects dataset, this is the data pertaining to the spheroids themselves. Again, remember these images were one spheroid per image. And so both of them are label 1 respectively, but these are separate images. Okay, so we have the volume and all that kind of stuff pertaining to these. Again, these are actually just duplicate spheroids just for demonstration purposes to show you that everything runs in batch. As we scroll across here, we of course have a breakdown of our channel intensities by the stratified volumes that we created from our spheroids as well. And as we get towards the end of our data set here, we have all of this intensity data. So what's really great here is we can get a breakdown of expression of proteins of interest as a function of kind of spatial distribution throughout the spheroid, right? So we now know the intensity, or the mean value, if you will, of channel 1 in the outermost band, because it's Q4. So these bands really give us the ability to stratify the shape and therefore query where inside the shape was something most or least intense. We also get the same for the halves as well. But right at the very end of this secondary objects or spheroid dataset, we get some summary values pertaining to the children nuclei that were found inside of it. Okay, so let's open these up so we can have a good look. Okay, so we get an absolute count of the number of nuclei found inside those spaces. Then we get a breakdown of the number of nuclei found. And again, these are all duplicate numbers because they're the same spheroid, just with different names for demonstration purposes. So we have a total number, and then we also have the breakdown of the number of nuclei and where they were found inside the tumor. So we see that we have way less in the middle and way more on the outside. And obviously these are not volume normalized numbers because our stratified bands are computed based on distance-wise measurements, because it's the solution that we came up with for this problem. But of course you could go ahead and volume normalize these as well, because you have the volume of each band saved as a record earlier on also. So we get to know which band contained the most nuclei, even as a volume normalized fraction if you so wish, as well as numbers of nuclei per entire spheroid. So as an example of how we can then start to get some really interesting insights into our data. And again, this is all fully supportive for 3D data sets. But yeah, that's a tour of the sort of the additional processing options we have here. And again, that's all been saved into my parameter file. So now if I wanted to redo this entire experiment, even with my additional secondary processing and such, I can reload these exact parameters and replicate that. I think that's all I had to show you. Thank you.